Alright, so today we're diving into one of the most intricate systems in our brain, the basal ganglia. It's a structure located deep within the brain, consisting of a group of nuclei, which are basically millions of nerve cell bodies working together to help control movement, behavior, and perception. The basal ganglia, or basal nuclei, lie deep beneath the cerebral cortex and are distributed across different regions of the brain, including the telencephalon, diencephalon, and mesencephalon, or midbrain. It exists in pairs, with one basal ganglia on the left side and one on the right side of the brain. Each pair is made up of two main structures, the globus pallidus, which itself consists of the internal globus pallidus, GPI, and the external globus pallidus, GPE, and the striatum, which includes the caudate nucleus and the putamen. Together, these form the core components of the basal ganglia. The basal ganglia connect with other brain structures, such as the ventral anterior nuclei and ventral lateral nuclei of the thalamus, as well as the substantia nigra in the midbrain. The overall function of the basal ganglia is to facilitate smooth, controlled movements by starting, stopping, and regulating motor activity, while also inhibiting undesired or competing movements. For example, when you walk, the basal ganglia help one leg to step forward while inhibiting the other leg, ensuring it stays stationary and you don't fall over. But that's not all. The basal ganglia also influence perception. Take a look at this classic optical illusion. You can either see a rabbit with two long ears or a duck with a beak, but you can't see both simultaneously. This is because the basal ganglia stimulate one perception while inhibiting the other, allowing the brain to focus on just one image at a time. Now, the basal ganglia receive input from nearly the entire cerebral cortex through what's called the cortico-basal ganglia thalamo-cortical loop, or CBGTC. This loop is a neuronal circuit connecting the brain's cortices, basal ganglia, and thalamus. It plays a key role in initiating movement, controlling skeletal muscles, and adjusting posture. Two main pathways govern this process: the direct pathway, which is excitatory, and the indirect pathway, which is inhibitory. This balance is crucial for maintaining smooth and coordinated movement. Two primary neurotransmitters are involved in these pathways: glutamate, which is excitatory, and GABA, which is inhibitory. Additionally, dopamine, released from the substantia nigra, acts on both pathways but has opposite effects due to two different types of receptors: D1 receptors, which are excitatory, and D2 receptors, which are inhibitory. In the direct pathway, the cerebral cortex sends excitatory signals via glutamate to the striatum. The striatum then sends inhibitory projections via GABA to the internal globus pallidus, GPI. Now, the GPI usually inhibits the thalamus, but when it's inhibited by the striatum, it can't continue its inhibition of the thalamus. The two negatives cancel out, and as a result, the thalamus is free to send excitatory signals to the motor cortex, initiating the desired movement. Essentially, the direct pathway increases motor activity. The indirect pathway starts similarly, with the cerebral cortex sending excitatory signals to the striatum. However, this time, the striatum sends inhibitory projections to the external globus pallidus, GPE, instead of the internal one. The GPE typically inhibits the subthalamic nucleus, but when the striatum inhibits the GPE, the subthalamic nucleus is free to send excitatory projections to the GPI. This then allows the GPI to inhibit the thalamus, reducing its excitatory output to the motor cortex, thus inhibiting undesired movements. Dopamine, released from the substantia nigra, has a critical role in modulating both pathways. When dopamine binds to D1 receptors on the striatum, it enhances the activity of the direct pathway, promoting the desired movements. In contrast, when dopamine binds to D2 receptors, it inhibits the indirect pathway, reducing unwanted movements. This dual effect means that dopamine facilitates smooth and coordinated movement. As an example, individuals using substances like cocaine or ecstasy experience abnormally high levels of dopamine. This makes the direct pathway for wanted movements overly active while suppressing the indirect pathway, leading to enhanced, sometimes uncontrollable, movements. Damage or dysfunction in the basal ganglia can lead to a range of movement disorders. For example, Parkinson's disease involves the degeneration of neurons in the substantia nigra, resulting in reduced dopamine levels and impaired movement initiation. Huntington's disease is characterized by molecular changes in the striatum, leading to uncontrolled movements. Wilson disease results from copper accumulation in the lentiform nucleus, which includes the putamen and globus pallidus, impairing their function. The basal ganglia develop from the embryonic forebrain, or prosencephalon, and later differentiate into distinct structures distributed over the telencephalon, diencephalon, and mesencephalon. Alright, as a quick recap, the basal ganglia consist of the striatum and the globus pallidus, working together to control movement. The striatum receives input from the cerebral cortex and communicates with other structures via the direct and indirect pathways. The direct pathway activates the thalamus, allowing the initiation of desired movements, while the indirect pathway inhibits the thalamus to suppress unwanted movements. Dopamine modulates these pathways, promoting smooth and coordinated motion. Thank you for joining this deep dive into the basal ganglia. For more comprehensive content on neuroscience and other medical topics, don't forget to like, subscribe, and check out King of the Curve for more educational content. Stay curious, and I'll catch you in the next video.